Have you ever imagined animals that can glow in the dark? What if we told you that there are cats, rabbits, and even cockroaches that do? This is possible due to a new type of technology called CRISPR Gene Editing. CRISPR Gene Editing allows the changing of genes by altering parts of a genome or the complete DNA of an organism. Genes are inside the cells of living organisms, and they contain DNA which instructs how the cells will carry out their functions. They are what makes us unique, as they decide our physical features and traits. With CRISPR gene editing, these traits can be altered into other traits.
But first, how did CRISPR gene editing come to be? It is actually based on the natural immune system that bacteria uses virus defense. This uses CRISPR-associated proteins or Cas proteins. When a virus invades a bacteria, the Cas protein acts as a two-in-one tool, a pair of scissors and a stitch. It first cuts off a segment from the virus DNA and stitches it into the bacteria's genome. These segments, called spacers, are placed in between repeating cycles of DNA or palindromic sequences. This arrangement summarizes what the term CRISPR stands for: clustered regularly interspaced short palindromic repeats. After stitching, the virus DNA is duplicated into RNA, which creates proteins. The RNA attaches to a Cas protein called Cas9, and they act as a team to search for a match to the virus DNA. Since the team contains a segment of the virus DNA, they will recognize that same virus if it invades again, prompting them to destroy it.
So how did scientists turn the CRISPR immune system of bacteria to a gene editing technology? They first designed the guide RNA that will match the gene they want to edit, and attach it to the Cas9. Together, they will search for the target gene and cut the DNA. With different RNA guides, scientists can edit various genes in any organism. When the DNA is cut, the cell will try to repair itself by using proteins called nucleases that connect the DNA fragments back together. This process is called non-homologous end joining. However, this can cause the gene to be unusable due to inaccurate repairs. Luckily, there is a process called homology-directed repair, where a separate template DNA sequence is added to guide the cell in repairing the faulty gene or inserting a new one.
Some examples of its current uses are turning pigs into organ donors, creating new treatments for cancer and blood disorders, and modifying human embryos.
CRISPR is not only limited to human genes. It can also be used for a variety of applications. But although we benefit from it, there's something we need to think about. This newfound technology raises ethical concerns. As we progress in the field of genetic engineering, we must remember that this tool is powerful. It has the power to help, but it may also have the power to harm.
There may be trait changes that are unplanned and potentially adverse implications that may arise from these changes made. All technologies have their pros and cons, but it is up to us to change the world.